All right, so today we're going to be talking about calcium and its role within skeletal muscle. So here, this is just a really good depiction of our neuromuscular junction, aka our NMJ. And so all that we are really talking about today is our calcium. So first thing that you want to see in this diagram is our motor neuron action potential. That's going to cause our calcium to come into the cell, which is then going to activate our acetylcholine release. So we're going to be paying attention today about how our calcium enters the cell and where does it come from. So this is like the step that we're focusing on today. Okay, before we get started, just want to point out that one motor neuron innervates multiple muscle fibers, but only one muscle fiber can have one motor neuron. So moving down to our calcium specifically. So this is a good depiction because we are able to see our muscle fiber action potential. So this is that spark, that action potential, and we see how quick it is. It's up and down. It's very quick. And then we see when our muscle actually contracts. So by the time that our muscle reaches peak contraction, our fiber has already been stimulated and over for a minute, almost 40 seconds, or milliseconds, 40 milliseconds. And we see down here, we have this latent period. And our latent period, this is actually a delay due to calcium entering the cell.
And that's actually calcium entering the cytoplasm, because the calcium is already in the cell. It's in either like the sarcoplasmic reticulum or somewhere else. So this is just the calcium entering specifically the cytoplasm.
So where does this calcium come from and why did it take a minute to get in here? So our muscle fiber is a lot more complicated, basically. So the big thing that we want to pay attention here that deals with our calcium is our sarcoplasmic reticulum. So this just kind of runs along our tubules and it stores calcium. So this is important because this is where our calcium is at, or one source of it, I should say. So if we go down here to this diagram, this has a lot. And with this, we just want to pay attention to kind of what does what. So we have our DHP voltage sensor. So that is going to start right here, our DHP voltage sensor. So this is a receptor that undergoes a shape change. And when there's a change in voltage, that's why there's these positive marks right here. So when there's a change in voltage, it's going to undergo a shape change. So that is then going to go to those junctional feet, which you can't really see in this picture here. It kind of skips over that. But it goes to these junctional feet, which then is going to promote a shape change. And once it promotes that shape change, it goes down here and it acts on our ryanodine receptors, which you can see right here. And so our ryanodine receptors, basically, it's once it has that shape change, it's going to cause open calcium channels. So this is what opens calcium channels. Now, what is this word, CICR? Or CICR is calcium-induced calcium release. So that just shows how kind of our next step, I guess you could say, because there are other forms of calcium in the cell that need more calcium to come out. But for this, it's just know that DHP voltage sensor acts on those junctional feet, acts on those ryanodine receptors to allow that calcium to come out of that sarcoplasmic reticulum. So now, what is this primary active transport or calcium ATPase? So this is how our calcium goes out of cytosol and into our sarcoplasmic reticulum. So that's just how it goes from the cytosol and into the sarcoplasmic reticulum. So this is that uptake when that contraction is over. And yeah,